And,
Let me dive in. I mean, I was fascinated by Dr. Goodenowe's book. I mean, I read something I did not know anything about, and I thought I knew a little bit about a lot of things. Something called plasmalogen, something like that, that he can measure, and he finds very highly correlated with, if not predictive, of various vessel diseases. So that will lead to heart issues and Alzheimer's. So let me tell you a little bit about Dr. Goodenowe. He does research into biochemical mechanisms of disease. This started in 1990. His curiosity about the biochemistry of life is insatiable today as it was 30 years ago. He's invented and developed advanced bioinformatic technologies, designed and manufactured novel supplements, and identified biochemical protomes of numerous diseases, including Alzheimer's disease and dementia, Parkinson's disease, multiple sclerosis, autism, ALS or Lou Gehrig's disease, schizophrenia, depression, and numerous cancers. I read his recent book, which is absolutely fascinating, and here he is to tell us more about it. Welcome, Dr. Goodenowe.
thank you so much, Susan, and pleasure meeting all of you. So by all means, bring your questions. I'm going to go through. We've got a big push on on our autism program right now, because we're launching a big protocol in autism, and we have that April's Autism Month. So I wasn't entirely prepared for this, but I do a lot of work on cardiovascular. So let's talk about this. So what I want to do today is I want to give you a little bit preview of the autism work that we're doing, and that's a live webinar that's going to be on April 29th. And then let's get into this myocarditis, ACE2 stuff, cardiovascular disease, cognitive impairment, death, and plasmalogens. Understand what these things have in relationship to each other. So my background, like Susan mentioned, is in psychiatric medicine. I'm also a synthetic organic chemist, focused on the biochemical mechanisms of disease, and you can check all that out later. And this is where I work, in Temecula, California. So we have a full institute here, ion cyclotron mass spec, magnetic sector, ICPs, really very advanced technology, a lot of work done in rare diseases as well as in aging. So the autism thing that many of you might be interested in, because of that crisis and the epidemiological explosion of autism over the last few decades, is just launching as well. So there's a course, I have educational courses on the website that you can look into, and the autism is free for the month of April. So you can jump in there and take a look at that. And then we talk about the Alzheimer's. There's a much deeper lecture for Alzheimer's. And then I do a lot of training on interpreting lab results in a context of longevity and health versus disease diagnostics. So with that, the autism thing, I won't spend too much time, just so that the slides are here for you to look at. There's 5 key lectures. There's a highlight lecture, mitochondrial insufficiency, microglial activation, the impaired myelination process, and then there's a protocol that's being launched in the next couple of weeks. We're having pretty dramatic results in the autistic population with our doctor network right now. We have about 1,000 doctors in our network. And so the highlight, we talk about the epidemiology, impaired myelination, which most people don't really get a lot of detail on, white matter development, neuroinflammation. Talk about the Cooper's-Zone model, show you some data on that today as it relates to cardiovascular disease and myelin. So mitochondrial insufficiency, activated microglia, impaired myelination. This is basically how it becomes clinically observable. And there's the protocol that deals with plasmalogens, myelin restoration, methyltransferase support, mitochondrial support, general nutrition. And so we can deal with the biochemistry of autism quite precisely. And then the recovery process is a bit more long-term. But in terms of autism, there's really the 2 slides to take home. Inflammation of the brain and impaired myelination are the core issues in autism. And there's extensive data on this. And that mitochondrial insufficiency is the primary underlying weakness and susceptibility that prolongs and exacerbates the acute inflammation. And ultimately, prolonged inflammation in combination with this underlying mitochondrial insufficiency results in a depletion of biochemical reserves and the establishment of a chronic inflammatory scenario that persists throughout the entire lifespan. You know, adults with autism have the same level of neural inflammation typically as they did when they were 8 years old. And so We talk about insufficient plasmalogen biosynthesis to deal with myelination of the brain. We have a lot of data on that. Certainly during inflammation events. Insufficient nutrition, and I'd be careful to tell people insufficient nutrition is not the same as malnutrition. But when the brain is inflamed, it goes through certain nutrients much faster than a normal brain goes through. And so they become depleted in certain nutrients, even if they're getting a good diet. And then once the autism cascade begins, it rarely, if ever, spontaneously resolves. So early restoration. And then the intervention strategy is really straightforward. The goal is to restore the myelination of the brain so we can restore normal neurological development in the child development, restore the methyltransferase capacity, restore mitochondrial capacity, and then some general supportive nutrition. So you can learn all about that. It's all available for you guys to go into. So on to the main event for today. Let's talk about cardiovascular disease, cognitive impairment, and plasmalogens. Why it happens, how it happens, and what you can do to restore and function and recover from these issues. This whole world of COVID is interesting because it gives us a large model to understand inflammation. Okay, because rarely do we have a situation where so many people have been exposed to common environmental stressors that have an inflammatory component to them. And so it gives us information that normally would be coming in little spurts. And one of the core components of it is that there's a difference between how a viral infection works. And we've been studying this since the 80s. So viral infections, so viral myocarditis is something that we've been dealing with for a long, long time. And so COVID is no different. There's nothing really special about it. The only thing special about it is its scale of which it's been, scale of human exposure to it. And so normally a virus, when it enters a cell, the purpose of a virus is to reproduce itself, right? It doesn't care about the health of the cell. The cell is just there to reproduce an entire viral particle and export it. And this is the exocytosis. And this viral particle is what we get immune responses to. And of course, the mRNA system was a bright idea, saying, you know what, let's not put the whole virus in. Let's just put the spike protein in through a messenger RNA program. And let's get the cell to make just a part of the virus, not the entire virus. And the hypothesis is that these spike proteins will create antibodies, and these antibodies for the spike protein will then be antibodies to an entire virus particle. So that's the theory. And so the problem is that, unfortunately, is that this kind of transfection doesn't hurt the cell so much. Cells are actually can be quite happy making these spike proteins, whereas a virus cell usually consumes the cell and it eventually dies. And the problem with the SARS-CoV-2 is that this ACE2 receptor is everywhere, right? The angiotensin converting enzyme is all over the place. And that's the tagline that gets the virus or the spike protein into the cells of our body. And again, this is not any different than any toxin we take. All toxins, whether it's glyphosate or whether it's paraquat or we use MPTP for Parkinson's disease, you have a toxin, and what happens is that toxin has selective activity on a given cell type. So when I want to create Parkinson's in an animal model, I use a toxin called MPTP. Now, it's a mitochondrial toxin, but what makes it selectively toxic to dopaminergic neurons is that it is selective to the dopamine uptake system. So the dopaminergic cells of the body selectively take up this toxin. So a lot of the toxicities that we study is really related to the targeting of the cell. If a toxin is a toxin, virtually in all cells, but the question is which cells get exposed versus other cells. And in this situation, we're dealing with the ACE2 receptor. And what happens is that regardless of whether it's the protein, spike protein, or whether it's the actual virus, each of them are active on this ACE2 receptor and create an inflammatory response. So we know we get this interleukin-6, just the spike itself. So the protein itself has an inflammatory component, independent of its immune concept of saying, hey, can it create antibodies? And these alarmins, interleukin interleukin-1 alpha. So it's a very specifically a pro-inflammatory cascade that gets created, and we see the cardiopulmonary effects of it. So the point is that it's the spike protein, by interacting with the ACE2 receptor, is independently inflammatory. So the point is that if your own cells are making a virus, like a regular virus, you get pneumonia, for example, or influenza, how does a viral infection dissipate? How does a viral program eventually get out of our bodies? And typically, what happens is we get this three-phase process. First, there's an infection prior to immune buildup. We get infected with something, whether it's a Coxsackie virus or whatever it is, and the immune system is not aware of it, doesn't know how to recognize it. And so these circulating virus particles, when you first get infected, they escape immune deactivation and they infect specific cell types depending upon the type of virus. And then the infected cell begins making these new virus particles. The infected cells, via exocytosis, I think someone's via exocytosis ejects new virus particles into circulation. And it's important to realize that the immune system is not activated against the virus producing cell, but it's activated against the ejected virus. And then the second phase of the viral infection is that our immune system begins to deactivate the circulating viral particles. So the circulating virus particles are now trapped by immune cells via virus-specific antibodies. And the rate of secondary infected cells begin to decrease. This is the critical component, right? We still have virus-creating cells, but the virus being spit out of these infected cells are being trapped before they do secondary infections. And eventually what happens is that the rate of cell death of our infected cells becomes greater than the rate of infection of new cells. So virus-infected cells, so you can't get uninfected. They either die via apoptosis, like the influenza virus, for example, or they become dormant, like a herpes or a shingles virus. But typically speaking, the metabolic demand of a virus infected cell is unsustainable. The cell will eventually die or become dormant. Remember, the virus is interested in its replication and propagation, not interested in stable transfection. And this is why when we do genetic engineering, and I have lots of decades of experience in genetic engineering of things, is that we never use a full virus infection. We only use a virus as a delivery mechanism for DNA, because so if we want to get a Roundup Ready plant, my goal is I use a virus to infect the plant with DNA that allows that cell to make a certain protein that digests glyphosate. The whole point is that the cell to survive the transfection. So that's why we don't use full viruses in genetic engineering. Anyways, so getting right to the point of what's going on here and why we can use this for cardiovascular disease understanding in greater detail is that COVID-19 itself, okay, not talking whether spike protein or not, just the vaccine, the viral infection selectively depletes this class of molecules, phosphatidylcholine BHP. This is plasmalogens, okay? So plasmalogens are dropped in the infection, and it gets dropped in the blood levels, it gets dropped in the lung. The plasmalogens versus, the polyunsaturated plasmalogens get significantly depleted upon COVID infections. And this is the omega-9 version, and I'll tell you more about that in a few minutes. But we know specifically of the phospholipids, and I'll tell you why it's so important for heart health. And we have data for decades on this thing, and that's one of the reasons why we see dramatic recoveries of people's heart function and lung function when we give them plasmalogen supplementation after COVID infections. But the point here is plasmalogen depletion is a big, big deal. Okay, this is what Susan was talking about in detail in the book relating to Alzheimer's, cognitive impairment, and mortality. But this is some of the data that we have from Rush University in Chicago, longitudinal trial, 17 years in the making. I have, I think, 11 or 12,000 blood samples from this study in my freezers. But this is taking a look at 1,262 individuals at 2 visits. And the difference between the 2 visits is 3.7 years. And this is the probability of having dementia based upon the plasmalogen levels in your blood, okay, in a very large population. So we're talking about people with high plasmalogens as a function of age versus people with low plasmalogens as a function of age. So a 90-year-old with high plasmalogens has like a 7% chance of having dementia, where it's a 40% chance if they had low plasmalogen. It's a very, very big number. And these are confidence intervals. And that we also showed that the probability of becoming demented. So what happens? So for here, there are 77 people with dementia at visit 1, and at visit 2, there's 226. Okay, we're dealing with an elderly population. Visit 1 average population was 81 years old and 85. So in the following 3.7 years, 149 new cases of dementia were observed in that 3.7-year period. The question is, can we predict which of those people got dementia and which didn't from baseline? And the reality is exactly the same. Those who had high plasmalogens at baseline had a very low probability of becoming demented in the next few years versus those who had low plasmalogens. So not only is it cross-sectionally associated, it's actually predictive of future cognitive decline. And when we look at the post-mortem brain analysis, so we look at people's brain levels in the temporal cortex, this is, very detailed cognitive assessment in this population over their lifetime and proximate to death. And this is the levels of omega-3. This is the DHA levels of plasmalogens. Arachidonic acid is here, so these are the polyunsaturated ones. And these are the brain levels relative to cognition. This is a log scale, by the way, on this side. And so you're dealing with this extreme difference in cognitive status based upon the plasmalogen levels, much, much stronger in the brain than it is in the blood, for example. So the more proximate we get to the biochemical mechanism of cognition, the more important. And the reason why this is so strong is that the vesicular fusion and release of neurotransmitters is dependent upon having very high levels of plasmalogens in your synapses. Of your ethanolamines, over 75% need to be plasmalogen related for full neurotransmission to occur. And that's why this correlation is extremely robust. And if we take a look at this in Parkinson's as well, because we have this large longitudinal cohort, there's less people with Parkinson's than with dementia, but still a pretty significant number. Again, we have these very, very large statistically significant associations on Parkinson's, whereas having high levels of plasmalogens reduce the risk by over half. It's much stronger than your age, actually. So your plasmalogens are more important than how old you are. And this is also done in Japan, so the North American population. So we've done this in multiple geographical regions around the world. So the associations, again, for ALS and Parkinson's disease, for this omega-3 plasmalogen is very, very robust, even in small populations. But what was really shocking about this story was mortality. And since in this population of 1,262 people, 557 of them had since died, since this trial has been going on for a long time. And so we were able to ask the question, what's the association with mortality? And we know the difference in age was 3.7, and then the average time to death for those who died was 1.6 years after their last visit. This is a probability of dying after correcting for the association for cognitive impairment. So being diagnosed with dementia already increases your mortality rate by 2.5 or more. But this is the mortality rate based upon your blood plasmalogen levels. And we're dealing with a 30-year differential in lifespan. So this basically graph says that people with a 95-year-old with high plasmalogens in their blood had the same 5-year mortality risk. They had the same probability of making it to their 100th birthday as a 65-year-old with low plasmalogens had of making it to their 70th birthday. Like, these are really, really scary numbers. And so it's more than just the cognitive impairment of the brain. Plasmalogens are really critical to many, many functions of the human body. And we don't have small amounts of them. We're talking 20, 30% of your brain. Over 50% of the phospholipids in your heart are plasmalogens. And that's what we're going to talk about here the rest of today, and why we're dealing with this crisis in some situations. And what happens is our plasmalogens actually continue to increase for a little while. Like our brain continues to myelinate. We actually continue to build plasmalogen reserves up until our 50s. And then it starts declining precipitously. Okay, so the age-related association, and if we take a look at very specifically the omega-3 levels versus omega-9, like the oleic acid is involved with your myelin sheath. The DHA is involved with your synapse and your neuromuscular junction for, say, sarcopenia and cognition, for example. But this relative proportion of plasmalogens has a much more dramatic age-related association. So this information, like there's very detailed, I go through the cognition, impaired cognition, talk about neurofibrillary tangles in Alzheimer's in the brain, talk about amyloid, deal with the ApoE genotype and how this affects cholesterol regulation, brain volume, oxidative stress, epidemiology. So there's extreme detailed information about all these sections in the book, but the lecture series actually goes into much greater detail, even more than what the book does. Now, when you come to cardiovascular disease, this is not new either. So this is 2004. This is the cumulative survival, okay, of cardiovascular mortality relative to the plasmalogen levels. So those who had higher than median levels, this was their survival plot. Those who had less than median levels, this was their survival plot for cardiovascular mortality. And again, the phosphatidylcholine plasmalogens, okay, are associated with myocardial infarction. This is the same one, the actual molecule here. They're talking about the omega-9 in this particular paper. That's the exact same one I showed you was decreased from COVID, the exact same molecular species. But this was obviously way before the COVID time. So plasmalogens, what the hell are these things, right? Why are they, causing such pain and disconcertion for us in our lifespan? Well, they're phospholipids. And when we think of the human body, the body is really trillions of cells, trillions of these cubes and spheres. And we think of these membranes as compartmentalization. It's what separates our heart from our brain from our lungs. It separates inside our cells. But we forget about the pure mass of it. So they're walls, they're biological walls, but they're also biological reservoirs of critical components. It's like having an electric car where they're using the shell of the car as your battery, for example. So the membrane structure of the human body is not just physical, it's a reservoir of things. And when it shrinks or modifies its composition, it affects many, many other things. And so they're basically made in these subcellular organelles called peroxisomes. But again, all of these intercellular organelles have membranes as well that separates it from the intercellular space. And so plasmalogens are made by a very special process in the peroxisome. And the trick with them is that we get very little of it from our diet, because the last step in their manufacture creates what's called a vinyl ether bond. And I'll show you what that means in a second. And you take a look at the membrane compositions and this is the cholesterol to phospholipid ratio, you'll see that the outer plasma membrane, the outer membrane of our cells contains a lot of cholesterol. Cholesterol is one of the most important molecules in human life. And then when you get on the intracellular cells, okay, the membrane around the mitochondria only has 10% cholesterol. So the compositions of your membranes change based upon their purpose and location. But the two main ones are these phosphorylcholines and phosphorylethanolamines. These make up the bulk of all of our membranes. And these are the two types of phospholipids that contain plasmalogen versions. Okay, the other ones, we have very little plasmalogen versions in the rest, but these ones contain high levels of plasmalogens. So a regular phospholipid looks like this. Okay, this is your biochemistry 101 phospholipid. It's on a glycerol backbone, got two fatty acids on it. And we make these willy-nilly. Okay, no big deal. We dietarily available. What makes the plasmalogen different is that at this SN1 position, instead of being a fatty acid, it's an ether. It's a vinyl ether. And this little bond here is like a fuse. Okay, it's designed to be exquisitely sensitive to acid. It is the main hydrogen peroxide neutralizer of the human body. Okay, we have glutathione. People talk about glutathione, they think about catalase, they think about these other functions. But pound for pound, your body makes plasmalogens like cannon fodder. You make a lot of it, and it uses like a fire hose. It just pumps it out, and especially the heart pumps the stuff out to neutralize the peroxide, which is one of our main issues with cardiovascular endothelial function. All of our cells and organs contain these things. Very high concentrations in the brain, heart, eye, lung, kidney. And it's this bond that makes them dietarily unavailable. So when you eat an animal food product that has plasmalogens in it, as soon as this bond hits the concentrated hydrochloric acid in your stomach, it just disappears. Okay, very, very little of it gets through the dietary tract. So we talk about COVID and the heart inflammation model. If we go look, this is not new stuff. It's 1983, okay? And we're dealing with leukocyte infiltration of the myocardium. It is observed histologically 24 hours after myocardial infarction, cardiomyopathies, and inflammatory myocarditis. Okay, so we're already talking about this 40 years ago, right? Leukocytes activated during the inflammatory immune process release cytotoxic processes. We know all this stuff. Okay, oxygen-free radical system mediate the cytotoxicity in the rat model of inflammatory lung disease. These oxygen-free radicals, superoxide anion, hydrogen peroxide, hydroxyl radical, okay, are excellent candidates for mediating leukocyte-induced cardiac dysfunction. And I'm going to show you some really good detailed information on this stuff. So we know what happened with COVID. We get white blood cell counts go up, neutrophils go up. Okay, so we do get this leukocyte elevation. And interleukin-6, relative minor versus severe cases, interleukin-6 goes way up. So it's very pro-inflammatory. And so we take a look at hydrogen peroxide and the hydroxyl radical activating this leukocyte depression of cardiac sarcoplasmic reticulum. And this is the same paper. We conclude first that activated leukocytes depress canine cardiac sarcoplasmic reticulum by an oxygen-free radical mechanism, that generation of hydrogen peroxide and the hydroxyl radical. And I'll show you why this is so related to our plasmalogen depletion that occurs, and why people in long COVID, we pull them out with plasmalogen restoration. So this, I think, is one of the most interesting studies in a long time. It's 2012, and they looked at the Coxsackie virus elimination in resistant and permissive mice. Okay, so here's an interesting situation where we look at the population distribution, where clearly the largest part of our general population can get the virus or can get vaccinated and have limited response. But there are subpopulations that get dramatically negatively impacted by either the vaccine or by the virus. And this showed how mitochondrial capacity determined whether or not someone recovered or not. And so the regulation is crucial for the viral elimination. So they had this virus-resistant mouse strain and this virus-susceptible mouse strain. And they looked at the mitochondrial chain, like the electron transport chain, so complex 1, 2, 3, and 4. And the virus-resistant, when it was infected, these complexes upregulated themselves. But in the virus-susceptible, it did not. And what was interesting was that the infected, these are the ones that die, okay, is that 90 days after viral infection of the Coxsackie virus, the myocardial cytokines, like this, 90 days after complex 1 of the mitochondrial change is still impaired. Like, so there's situations that happens where when someone gets infected, they get knocked down and they don't get up. And this is very interesting how just mitochondrial capacity determine the difference between two mouse strains and their mortality for the Coxsackie, specifically myocardial infection rates. And this was very, very correlated to the complex 1 activity. And so A high rate of cardiomyocyte apoptosis supports the functional recovery of myocarditis patients by assuming an efficient elimination of infected and damaged cells. Okay, we got to get rid of the infected cells once they get in. So again, this is this plasmalogen that I showed you in the earlier slide that is depleted in COVID. This is where it gets pretty interesting. So again, we're not talking earth-shattering last month data. We're talking 1989 data here. And it says here, the predominant phospholipid classes present in canine myocardial sarcolemma were choline and ethanolamine glycerophosphate, which contain 47% and 28% respectively of total lipid. 54% of the choline pool was plasminylcholine. 67%, this is in the heart, 67% of the ethanolamines are plasmalogens. Over 50% of the membrane phospholipids, okay, in the heart are plasmalogens. And we know that oxygen, I told you this already, is known to rapidly add to this vinyl ether bond, and it basically neutralizes free radicals. So furthermore, since free radical oxidation of myocardial lipid constitutes occurs during myocardial ischemia and reperfusion, we demonstrated that oxygen-mediated oxidation of plasminylcholine resulted in the generation of several products which have chromatographic characteristics. Basically proved the point that in the myocardium, the oxidative stress is depleting or reacting with the plasmalogens and reducing them. Which leads me to a short story on oxidative stress itself, because we talk about, because peroxide is really critically understanding of endothelial dysfunction. But we forget where this comes from. These are these four complexes, 1, 2, 3, 4, right? These are the complexes of the mitochondria. I tell people the human body is a hybrid electric car, right? We burn hydrocarbons to charge a battery. This is a battery we charge, and this battery runs our ATP pump. Okay, so we run on an electrochemical battery, what humans run on. And we create this ATP, which is our energy. But we run through these four chains. People hear about coenzyme Q10. This NADH is human electricity. It's the first step in the irreversible combustion of hydrocarbons. Once NADH is created, it cannot be put back in the box. It's like electricity. It has to get used. And typically, NADH has to get rid of its electrons through the electron transport chain. The first step is it adds to CoQ10, right? Which is why people talk about CoQ10 a lot. So the very first step is it transfers its electrons from the ubiquinone, and it gradually becomes ubiquinol, which is actually pro-oxidant. And then it goes to cytochrome C, and this is where our critical elements like iron and copper, you'll see them here. So the electrons from ubiquinol now gets transferred onto these iron and copper molecules. And this is where it basically takes oxygen. We're basically reducing sunlight energy back to heat energy. And so the iron and copper grab the oxygen, pull it apart, and then meet these two waters. Okay, this is the safe way of converting oxygen into water. That's what our bodies are supposed to do. But anything that doesn't get converted properly through this process turns into what's called the superoxide anion. And so anytime this gets overwhelmed, we generate a whole bunch of this superoxide anion. And the biggest thing that drives superoxide anion is NADH. Okay, NADH is the electricity that does that. And we get this superoxide anion. Now, when a cell gets stressed, cardiac cell, lung cell, brain cell, doesn't matter what cell it is, if the intracellular energy cannot convert NADH to energy, it spits these electrons out through these NOx enzymes. Okay, it spits them out. So you know what? Let someone else deal with them. Spits them out into the extracellular space, and you get these extracellular superoxide anion. And this normally gets converted to peroxide. And the superoxide process is very robust. Typically, it's an inducible enzyme. And once we get this peroxide, it is a very powerful signaling molecule, but it's only supposed to last for a very short period of time. This peroxide gets reduced by catalase, glutathione, and plasmalogens into water. If it doesn't, and so this is where catalase, where your iron levels, and these are all things that are associated with poor inflammatory outcomes. People that are anemic have very poor outcomes when it comes to these infection rates. And then the last one is glutathione. You've seen this 1,000 times. So glutathione is the free sulfhydryl. It works with selenium and riboflavin. These are these core critical nutrient elements. If you're under oxidative stress, these things get consumed. But glutathione is a very powerful peroxide reducer, and this is how it works. NADPH goes to NADP+. Again, this is all related, this NADH or NADPH. And plasmalogens are complete neutralizer. This is like pouring water on a fire, is what plasmalogens do. They completely neutralize it into water and a hydroxy fatty acid. Totally benign outcomes, which is why your body uses it so powerfully. So this is what happens, how it's supposed to work. You have many, hydrogen peroxide, the body has many ways of trying to get rid of it. It says, you know what, we're not going to keep this around very long. But if we don't neutralize the peroxide, this is what ends up turning into the hydroxyl radical. And this is where oxidative stress really begins in the human body. So, and this is where you get membrane peroxidation. So from hydroxyl radical to lipid peroxides. The reason I'm showing you all this is because people measure malondialdehyde levels. You want to know why you're measuring them, where they come from. So when you get this hydroxyl radical caused by the incomplete neutralization of peroxide, period, full stop, that's where it comes from, it will react to a polyunsaturated fatty acid, typically in your membranes. And you get this lipid peroxide. And this lipid peroxide, a molecule, a biomarker of membrane peroxidation, is a simple little molecule called malondialdehyde that you can measure in a blood sample, whatever you want to do. And we bring this up in autism because malondialdehyde levels are elevated in autism. They're also elevated in COVID, okay? So MDA levels are significantly elevated in COVID. All these oxidative stress situations do that. Here's another one where the endothelial and oxidative stress markers. So hazards ratio was 3-fold for the malondialdehyde levels. So people that had higher levels, and it was significant, that elevated malondialdehyde had a 3-fold increase in death rates from COVID infections. And this is the survival rate of people with lower malondialdehyde versus those with higher malondialdehyde. Okay, so this is pretty robust information. And this is the malondialdehyde levels in COVID recovered versus COVID deceased patients. Okay, in that there is a significant correlation with the survival rate based upon the level of oxidative stress markers, and specifically lipid peroxidation. So back to the heart. Okay, we're here to talk about cardiovascular disease. And fundamentally, endothelial dysfunction is cardio and cerebrovascular disease. That is what it is, right? You get endothelial dysfunction, leads to atherosclerosis, coronary artery disease, peripheral artery disease, parotid artery disease, heart. This is where it all leads from endothelial dysfunction. And it gets more severe at the more severe dysfunction that occurs. And when you look at the vascular morphology of your vascular system, okay, this is what it looks like. And this is where the relaxation and contraction capabilities come into play from a cardiovascular perspective. And what happens is endothelial injury dysfunction creates breakdown of this endothelial barrier. And specifically, LDL gets in. You get macrophage uptake. You get these foamy macrophages, okay, oxidative lipid peroxides, and this will eventually lead to this process, right, where you have this evolution of atherosclerosis, where what's happening is this lining gets inflamed and it gets spreading, so it's squeezing it. So we don't have actual buildup on. It's not really building up; it's squeezing it. Your vascular system gets squeezed, by this inflammatory component, and then you can get a rupture, which of course is not a good thing. So this is reduced blood flow versus a rupture. And if you have a blockage, okay, you get ischemic stroke. If you have a breakage, you get hemorrhage stroke, right? Most people, when you get a stroke situation from a cardiovascular event, it's going to be ischemic. It's from a blockage of your vascular system. Point is hydrogen peroxide drives endothelial cell leakage. Okay, this is a very good paper that looked at arterial endothelial barrier dysfunction, and they used homocysteine, but looked at the hypoxanthine xanthine oxidase free radical generating system. And so what happens here from the purine system, we get the hypoxanthine, xanthine to uric acid, and this creates oxidative, this is an oxidative stress pathway, but it fundamentally creates hydrogen peroxide, which is supposed to be neutralized by catalase. And what they showed was the equilibration of albumin. So they're looking at albumin transfer across monolayers of the aortic. They're looking at a very good function of this. And if you just provided the hypoxanthine, not much. The enzyme, not much. But if you put the two together, okay, then obviously you get this huge endothelial dysfunction graph. And what was interesting, this is the endothelial dysfunction caused by this peroxide forming process. And if they add increasing levels of catalase, completely neutralizes this. A very, very elegant work. I urge you all to read this paper is really great because it shows really simple cause and effect situation. Then what they did is they looked at in greater detail other things. So is it just peroxide, or can superoxide dismutase do the same thing? But no, what they found was it was hydrogen peroxide. Basically, and I can't say it better than what they did in their summary. It said, endothelial barrier function was assessed by use of an in vitro model which transfer of tryptophan, blah, 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 across the monolayers of bovine aortic endothelial cells. Addition of either hypoxanthine or xanthine oxidase alone during 90 minutes did not affect the albumin transfer, but a combination did. The increase in albumin transfer was abolished by catalase, reduced by allopurinol, but unaffected by superoxide dismutase, hydroxy radical scavengers, and so on. Made it very clear that hydrogen peroxide itself increased albumin transfer across endothelial cell monolayers. So really wonderful paper. And so what happens then, if you take a look at what it does, it'll antagonize the activity of nitric oxide, which many of you will know if you're looking at heart disease. So here's a good paper. They're focused on rosmarinic acid, but that's not the point here. The point is that they did a really good study on peroxide and the endothelial cell layer relaxation and dilation function. And they showed basically very clearly with the acetylcholine that hydrogen peroxide at increasing doses dose-dependently reduced the vasodilation rate. Okay, very, very clear, very robust. And then they looked at the relationship between endothelial and nitric oxide. And so L-name is an endothelial nitric oxide inhibitor. And so it showed here that the relaxation rate, if you inhibit nitric oxide, okay, it reduces the relaxation rate. So nitric oxide is good for you, clearly. And if you add peroxide here, it also is worse. But if you take peroxide and you add the nitric oxide, you get a doubling of the effect. So again, nice, simple, elegant, good research. So lipid peroxidation, atherosclerosis, cardiovascular disease, myocardial infarction, and diabetes. So these things, this uncontrolled superoxide peroxide basically ends in death. We get microglial activation, neurodegeneration, demyelination. Don't have time to go through these details, but this is also very well detailed science. Lipid peroxides and atherosclerosis. This was a good paper, again, years and years ago, showed the association between atherosclerosis. So lipid peroxides are elevated in ischemic heart disease, okay? and peripheral artery disease. But they're correlated with increased triglycerides, not cholesterol. I don't have time to get into the whole cholesterol thing today, but that's a big red herring in this field. And the high triglycerides are a symptom of underfunctioning peroxisomes. But this is lipid peroxides, okay? Clearly, cholesterol, triglycerides are elevated, and lipid peroxides are elevated. I won't go through it. So this association is very robust. And it's associated in the aortic more so than in the pulmonary aspect of your vascular system. So cardiovascular disease, lipid peroxides are elevated, both in the aortic and in the pulmonary system. And the progression of coronary artery disease from stable angina towards myocardial infarction is also associated with the level of oxidative stress. If you take a look here at malondialdehyde levels, where am I here? Malondialdehyde as the stable angina versus unstable angina, the correlation between the difference between those two is extremely higher levels of malondialdehyde in the unstable angina. Same thing with stable angina versus myocardial infarction, this oxidative stress thing, which makes sense because we know this is coming from this endothelial dysfunction process. So lipid peroxides correlate with the diabetic cardiovascular outcomes. And catalase. Catalase is decreased. So when lipid peroxides are out of control. I'm going to show you. So the point here is lipid peroxides as a fire hose. And I'm going to skip through this part. Told you already, 53% of the cardiac sarcoplasmic reticulum is plasmalogen. And this, I'm going to put these slides for you guys so you can look at them, but this shows you how the heart actually does this. It uses phospholipase A2 to pump plasmalogens out of the membrane. This is like having a town with a reservoir next to it. And you get that reservoir is being fed by a stream made from the mountains. And then you get a fire in town. Fire trucks come in, they pump a hose in the lake, and they start pouring water on the fire. That's fine if you go through half your lake and the fire is out. And then the lake can take some time to build up again. But if that fire goes on too long, or if you run out of water, you're screwed. And this is basically what's happening, and the detailed science of this is really robust. But I'm going to, just in the interest of time here, it shows how the plasmalogens, this is the alkyl glycerols, are elevated and the diacylglycerols decrease. Membrane-associated phospholipase A2, again, this is well-established science. These guys have been working on this stuff for years. And I'm going to just show you how it has a selectivity of the plasminylcholines, and so on and so forth. So I'm going to, just in the interest of time, I'll leave you guys these slides, but I want to show you what happens when we actually, this is just really good science that these guys did. And bottom line is, this is why plasmalogen precursor therapy has such a dramatic effect. Like, we pull people out of ICU units in days. It's just really quite crazy. So when they have this myocardial problem and they get the plasmalogens endogenously, like or supplementary wise, they just suck it up. And the reason why this works is all been detailed in a very, very good way. Okay, I'm going to go past that. And I told you this with the interleukin 6 and the white blood cells. Phospholipase, those people who survive COVID, they're actually able to phospholipid, they actually have more. They can suck more out of their membranes. And they have more reserve capacity. It's like, this is an indication of how much water is in the lake before the COVID infection, this particular study. It's pretty cool. Okay, so let's talk about plasmalogen supplementation. MPTP, I showed you a little bit before, it selectively impacts the mitochondria. We use it for animal model for Parkinson's disease. And it selectively hits that complex 1. Remember the Coxsackie virus slide where it left forever. It was the complex 1 here that stayed down. MPTP, active gradient is MPP plus, hits complex 1. And it selectively, MPP plus, elevates this in a dose-dependent way, the hydrogen peroxide levels. And in the brain, you get this massive microglial and astroglial inflammation in the substantia nigra pars compacta here. As you'd expect, we see this in autism, we see this in other neurological inflammatory diseases. And this is from the activated microglia. I talked about that, but this is what it looks like. Look at how nasty these things are. So the control IBA1 is for microglia, and you can just see this is just, you don't want that in your brain. My goodness gracious. So what happens is, if we take plasmalogen precursors and we give animals plasmalogen precursors prior to giving them MPTP, this neurological toxin also drops your plasmalogen levels in your blood, just like we saw with COVID, just like we saw with other things. But if you give the plasmalogen precursors, you prevent that serum depletion, but you also completely prevent neurodegeneration in the brain. Both the striatal dopamine transporter as well as the vesicular monoamine transporter. And if you take a look at an acute stroke, focus on this cardiovascular space, a 7-minute ligation model of stroke, looking at the hippocampal denervation here, like the loss of neurons. The point here is going to be not this, but the fact that this IBA1, which is a marker of microglial inflammatory, it just goes crazy, right? So a stroke model, 7-minute, you're looking 10 days and 30 days later, you get this massive induction of microglia. And this microglia goes up crazy. So this inflammatory part of the brain. And this microglia basically attached to, the red here is myelin basic protein. So this is your axons. The green here is your microglia. And you'll see it just engulfs these things and just basically eats them. And this is the astrogliosis, which is another story that's actually trying to help you. And it goes on. The point is that you lose this myelination. This is a key thing for autism research as well as aging. I have another presentation to do with white matter and aging as well. But this happens, we studied this white matter loss in a model called Coopersome. And this is what Coopersome does. It does exactly the same thing, this astrogliosis, microglial inflammatory. Well, if we treat animals with Coopersome, okay, you end up with this very reproducible, it's been studied for 50 years. So Coopersome's been around for a long time, for myelination of the brain, for multiple sclerosis. We study for schizophrenia, all these white matter diseases. It's a wonderful model. It's been robustly studied, but very reproducibly reduces white matter, causes white matter demyelination of the brain over a course of time. But it also depletes plasmalogens, and it depletes the plasmalogens prior to the demyelination rate. And what happens is, if you take the 6-week Coopersome study and say, what happens if we treat these animals with plasmalogens? Okay, dose dependently, 25 milligrams, 50, 100 milligrams. These are reasonable doses, by the way. I'm personally on around 50 to 100 milligrams per day, per kilogram. And the plasmalogen precursors that we use are basically designed off of human breast milk. So human breast milk has high levels of these plasmalogen precursors, which are important for children's myelination early on in development, as well as the mother's placenta, for example. But anyways, this is orally bioavailable. It passes the gut, it converts into this alkyl glycerol, and then it restores plasmalogen levels. And when you do that, you prevent the destruction of plasmalogens in the blood. You maintain plasmalogens in the cortex of these animals. You maintain them in the cerebellum of these animals, and it completely eliminates any Coopersome demyelination. So this is what a regular diet, this light staining is all your myelin, and it disappears here. And this is what it looks like when you have the plasmalogens, a complete restoration of the myelin of the brain. And I'm just showing you what we're looking at is these guys do a better job making pictures than our lab does. And so this is what it's supposed to look like here. And then this is what Cooper's own does with a better myelin stain color contrast. And so that's the difference between here and here. This is what Cooper's own would normally look like. And this is what it looks like after plasmalogens. And in fact, if we give them plasmalogens even after they've been demyelinated, they remyelinate. Okay, we can restore the myelination. So this is 7 weeks on Cooper's own, and just the last 2 weeks to go on plasmalogen. So they had 5 weeks of demyelination. Cooper's own is still being given to the animals every day, but for the last 2 weeks, they're given plasmalogen precursors, and you get myelin basic protein being restored, CNP being restored. So in humans, okay, we've been doing this now in humans now for several years, and this is why the trial designs are so robust. We did an escalating dose study with Dr. Jordan in Santa Monica on the omega-3, the DHA plasmalogens, from 900 milligrams to 3,600 milligrams per day. And we get this dose-dependent increase in plasmalogens in the blood. So if you take the plasmalogens, your blood plasmalogens go up. They go up a little, they go up a little more, they go up more. And so it is a dose-dependent curve, as you'd expect. But this is what happens to malondialdehyde. Okay, as your plasmalogens go up, you totally suck your malondialdehyde down. So these are the individuals that had baseline malondialdehyde levels way up here. Every single one of them had normalized malondialdehyde within a few months. Okay, in fact, it happens much faster when we do short-term studies. And catalase, remember how I mentioned catalase and its importance to reducing hydrogen peroxide? Again, if you increase the plasmalogen levels, you improve catalase function. And same thing with superoxide dismutase. People at low levels get normalized, people at high levels, because superoxide dismutase is a goofy enzyme. But we actually found cognitive improvement. These are clinical dementia rating scales of 2. So this is moderate dementia. This is not mild dementia. This is moderate. Okay, 75%, 3 out of 4 of the people that had moderate dementia improved by an entire CDR score or more within 4 months. Within 3, like this is so it's not stabilizing their cognition. We're talking about improved cognition, and their mobility using the sit-stand rate, the neuromuscular function dramatically improved as well in a very short period of time. And both, even though it's only a 22-person study, each person being their own control at baseline, the cognitive improvements and the mobility improvements were statistically significant. And the take-home message is all of this. Okay, plasmalogens are part of this membrane. Okay, they really are an antioxidant fire hose. They involve for myelination, does many things in the brain, like its synaptic function, but the oxygen gets converted to superoxide anion, gets converted to peroxide. This peroxide cannot be allowed to persist. Okay, and this is the nexus point of cardiovascular disease. And if you can stop the conversion of peroxide into the hydroxyl radical, all of this cascade that happens afterwards gets stopped. And this is where I leave at the end, because we talk about autoimmune disease and multiple sclerosis and autism, and it's kind of a fallacy. Okay, inflammation is supposed to be attracted to damaged cells before they go into lysis, and they're supposed to be there to clean up and isolate the debris so the debris does not spread. Otherwise, you have the equivalent of sepsis, but at a cellular level. So those slides that I showed you with those microglia engulfing the axon, that's actually supposed to happen. Okay, as far as the microglia is concerned, that is a damaged cell. It's got peroxidated membrane. It's getting ready to go into apoptosis, and we don't need all of that intracellular crap all around the body. It's supposed to be there. It's supposed to deliver the final death blow. However, the immune system is not attracted to healthy cells. So once the chemoattractant signal is gone, the immune systems dissipate and become dormant. And this we see with multiple sclerosis, longitudinal studies in multiple sclerosis. Once the immune cells are gone, new cells can replace the old and restore function. Only the isolated damaged cells are supposed to be affected, not large regions of cells. So these immune cells are designed to induce redox failure. Didn't go through this, but that's how they do it. They use a process called glutamate excitotoxicity, and they also do cysteine starvation of the target cells. But if healthy, undamaged cells around the damaged cells become damaged during the immune response, then they create new chemoattractant signals and perpetuate the immune response. And multiple sclerosis and autism are examples. So what we call autoimmune is really just mitochondrial insufficiency, and it creates this spread. So viral and related myocarditis cause mitochondrial stress. Mitochondrial stress in excess of the person's resolving capacity results in the propagation and depletion of membrane plasmalogens. And so endogenous antioxidant mechanisms, mitochondrial, plasmalogen reserve capacity, plasmalogen biosynthesis, determine the mitochondrial stress load that a person can handle. And that's why the plasmalogen supplementation has such dramatic effects. It's just like it's bringing in water in tanker trucks for people. So that's my long, I try to go through it. I wasn't really expecting to do a full cardiovascular speech today, but hopefully that is of interest, and I can stop sharing and we can discuss at leisure. So I apologize for being a bit long-winded. I try to cover some stuff, at least give you guys a slide deck for your interests going forward.
Okay, so what people can raise their hands for questions or put the questions in the chat. The only questions I can see are the people who have their picture on. Otherwise, last time I couldn't see a lot of people who had their hands up, just didn't show up anywhere. So Alfonso, why don't you start?
Hi, Dr. Goodenowe. Thank you so much for the talk. I just had a quick question.
Sure.
Does your institute have any timeline of the sort of this metabolic normalization process that you're gonna do, or?
Oh yeah, so we typically, the biochemical normalization usually occurs within weeks, but depending on how aggressive you deal with it, but the plasmalogen restoration can happen. Like, so for example, in children with autism, we start seeing symptom recovery when we get the neural inflammation down. Usually within 2 weeks, we start seeing some positive outcomes. And then it just takes a slow reversal point past there. But say C-reactive protein levels, we can reduce CRP levels in 24 hours. Like 24 hours after, if you take a high dose of the neuro, you'll drop your C-reactive protein levels the next day. So it's pretty fast. But for overall metabolic normalization, so these biochemical intermediates are designed. So fixing the biochemistry doesn't take that long, a few weeks, as long as people can take it in. And the next question is recovering from whatever accumulated damage has occurred, right? Sometimes we confuse curing a causation with recovering from a disease. And I tell people it's like scurvy, like I can replace the vitamin C pretty quickly, but that doesn't mean that the person's going to, all the sores and things are going to recover the next day. Like the person has to recover now from the accumulated damage. And so those are the two different processes that we deal with. But yeah, metabolic normalization can happen pretty quickly.
Yeah, no, I was speaking more of I think you've mentioned in the past that you're planning to take clients inside of the institute and sort of investigate with functional MRIs and other things, and maybe do IV or other services like that. And I think you are now focused on paroxysmal diseases, but I was just wondering whether this is something you're still planning to do for other people.
Oh, we're doing it already. I have over 1,000 doctors using our program. Like this is widespread already. So yeah, so doctors go in, they get certified, they use our supplements, but they can use others. Like like a lot of these things are dirt cheap, right? We talk about N-acetylcysteine to restore your glutathione and sulfur amino acid levels, and carnitine function and CoQ10. So some of these things are it's not, the plasmalogens are a big hammer. They contribute a lot, and it's one of the hardest things to get into this. Can't get it nutritionally. But when we do metabolic programming for people, it's we do the advanced blood testing every day, and we teach people how to restore normality. So yeah, that's going on now. That's not a future thing. That's going on right now.
I have a question for the layman in the audience. Can you explain in a very simple way so we can repeat it to others so we can spread the word what a plasmalogen is and how to increase the levels and maybe, describe that you do have a test for it. Clearly, you showed that it's very highly correlated, if not causative, on endothelial function improvement.
Yeah, so plasmalogens are fundamentally a membrane phospholipid, and they have 3 or 4 main functions. They are for the myelin sheath, so your Schwann cells for your peripheral neurons, and your oligodendrocytes for your central nervous system. That's your key molecule for maintaining myelination of the brain. And then that's one main role. The other main role is in the synaptic function, so the neuromuscular junction, the release of neurotransmitters, and the neurotransmitters in the brain. That's the other critical function. The third function is membrane structure, like in terms of cholesterol regulation. It's really related to your HDL and reverse cholesterol transport system. So your HDL system and getting rid of cholesterol out of your foamy macrophages. And then the fourth main role is going to be the pure antioxidant capacity, just actual pure fire hose neutralizing peroxide. And so, yeah, we can measure them in blood. And then you have 2 different types of precursors. One is the omega-9 that we designed called protomglia. That's for the white matter tracts and for reducing neurological inflammation. And then protomneuro has pure DHA plasmalogen, and it's designed to enhance the neuromuscular junction and synaptic function. So that's in a nutshell, that's what plasmalogens are. Your body, from a natural perspective, people that have good HDL levels and low triglycerides, like if you build skeletal muscle mass, those type of things that are healthy for long-term life, those individuals will typically have higher plasmalogen levels. So there are ways of keeping your levels up from a lifestyle perspective. It's just hard to maintain it long-term.
And what is there any, is there any, what supplements would you recommend? I mean, NAC, a lot of them, or is there some particular?
Well, those are for your mitochondrial health. So for the plasmalogens, there's really no choice, but you take the precursors that we've designed. Okay, that's why they were made in the first place, because there isn't any natural source. There's some in shark liver oil and human breast milk are really the 2 natural sources of plasmalogens that you can find. Human breast milk, of course, comes it's a good thing. Shark liver oil has a whole bunch of other things with it that I wouldn't recommend huge doses of, but it's out there. And then those are your main endogenous sources. And that's why we designed, pure precursors. It's like kind of L-DOPA for dopamine, for example. That's what we do. It's a plasmalogen precursor designed to be absorbed, go into all the cells of the body, and allow those cells to replenish them. Then the other nutrient is really basic stuff. Like we want to make sure the mitochondrial function is in full capacity. And those are your good old classic B vitamins. We forget about niacin and riboflavin. Sometimes people don't take those things seriously enough. And because if you're in inflammation, your body burns that stuff up. Getting sulfur amino acids, so especially vegetarians, make sure they keep up on their sulfur amino acids. It's hard to get the sulfur amino acids in plant-based protein sources. So these kind of things that, so if people get depleted, so there's lots of things. There's basic stuff though, but for the plasmalogens, you really are stuck with these precursors to get them up.
Do you have a supplement formulation to make this easier for us? Because
yeah, that's what it comes in a bottle. It's plasmalogen precursor. So it's protomneuro and protomglia are the 2 supplements. They come in gel caps and in a liquid formulation.
So
our kids
do we find those on www.prodome.com? Is that where people can find it?
Absolutely.
Okay.
And then the blood testing as well, and then the education and training for doctors is on there as well.
Okay. And how does this interact or correlate with the glycocalyx?
That's a good question, because getting that really nice fine hair absorption, especially the blood-brain barrier, is critical. I don't have any direct knowledge. I have no direct interaction, but I think that the importance of the glycocalyx over the last several years is interesting. It's a cool thing. I don't know the exact answer for that one. I don't have any before and after effect. I would assume if you improve membrane structure, reduce oxidative stress in those things, you're going to improve the glycocalyx. But I don't have direct.
Maybe Anna might want to talk a little bit about that. Anna, are you interested in mentioning that? Well, before Anna comes back, let's answer Atta's question. So Atta, if you can unmute yourself.
Great. Well, it was definitely a very informative seminar, and I'm glad to follow up with the doctor more in details. A couple of questions that I have is, what type of inflammatory biomarkers do you think we could apply in order to see the effect or the cause of this reductions or the redox? Now, we're designing a new test for testing for TNF alpha and 20 different biomarkers. I was wondering if you could give recommendations of what type of inflammatory biomarkers we should do.
I think
And also, what is? Yes, go ahead.
But so when we look at metabolic function in a blood test, the simple marker, like so malondialdehyde is a good marker, okay, for an overall oxidative stress. You don't get malondialdehyde if you don't have peroxidated fatty acids. So that's a good end product right there. The other one is uric acid is a good one. We don't, uric acid is probably the best marker of high NADH levels. So when uric acid levels get above 6.5, you know you are in a high NADH excess. So that's a good marker as well. Low uric acid is also a big problem because that's a measure of NAD deficiencies. So uric acid, malondialdehyde are really good, simple markers. And we look at phospholipid ratios, the plasmalogens themselves, and we look at mitochondrial function, like we can measure the acetyl-CoA leakage out of the cells. So we measure fatty acid elongation markers. That's another very powerful marker. And we look at peroxisomal function. We can measure product and substrates of peroxisomal beta oxidation in a blood sample. So there's other things that we do collectively. So the proton scan blood test incorporates a bunch of things. They're designed for doctors to look at an individual's health and understand the peroxisomal function, mitochondrial function, nutritional capacity of the individual, phosphatidylcholine, those things like HDL levels. But for oxidative stress, your interleukin-6, your malondialdehyde, liver peroxides, those are usually your best ones because those are really related to the end product of that. But
this training is,
and C-reactive protein.
Sorry.
Go ahead.
Are these trainings only available to physicians or any kind of care providers?
Yeah, any kind of healthcare practitioner. So it can be registered nurses. Basically, the educational courses are available to anybody. The roundtable, so we have two models. That we have a doctor network where doctors become certified. There's a roundtable we meet every month on, and they share case studies. So our advanced studies, say in ALS or Parkinson's, and so people will share different case studies. So now I'll do a detailed lecture like this once a month on a given topic. Like I'll give one on bipolar a couple of weeks ago. I gave one on autism and the cardiovascular and so on and so forth, dementia. And then the doctors will be practicing. So we have all these doctors out there treating patients now, and then they will share. So that's a private group that doctors can share case studies and build better and better knowledge. And then we have user groups like our autism, like patient groups that we help support so that patients themselves have a mechanism, because we do this rare disease in children and now with autism, there needs to be a place for actual patients. So yeah, so the answer to the long answer to your question is on the website, you can, any kind of doctor or practitioner, if you will, even if it's a nutritionist, for example, can become part of the roundtable and be included in that kind of conversation. There's education and training. And then they also get on our referral list. So there's a map that people can find a doctor near them. And then that's kind of how it goes.
Good. Thank you very much.
You're welcome.
Let's see, there's a question in the chat about recommended supplements if somebody has bradycardia That's a slow heart rate with a pacemaker.
I don't know exactly. I think we look at their general health. Okay, there's certain things that are just important for everybody, right? So when I look at their blood work, like the basic rule of thumb, if you look at holistically the biochemical profile, you want to get their fasting triglycerides under 100. You want to get their total cholesterol somewhere between 200 and 240. As soon as it gets under 200, all-cause mortality starts doubling. So you got to get those cholesterol levels up. You want the HDL over 50, ideally somewhere in that 60 range. I really worry about people with low creatinine, so muscle wasting. So make sure you see people with creatinine levels down at 0.6 or 0.8. So you're in the muscle wasting space. The heart is a muscle, so you can't get low on your creatine levels. So creatine supplementation is something so many people should be on as a general rule. Look at their phosphatidylcholine levels because you're reverse cholesterol transport. So the quality of the membrane structure. So all of these things come into play. So the general health model, you want to make sure these markers are where we are. We know exactly where they should be, right? Want to make sure that uric acid is not over 6. Keep it between 5 and 6. And we can move out, we can adjust all these biomarkers accurately. And then when you get health up, you can start juicing, like you want to increase heart rate, the omega-3 plasmalogens are good for performance enhancement. But fundamentally, heart's a muscle. The system needs to be is designed to work. We know the parameters that we're supposed to be at, and then you can just kind of go from there. But I wouldn't have a specific protocol for that.
There's a discussion in the chat. I mean, for the vegetarians, they're talking about clary sage seed oil. Do you have any comments on that? Because the ratio of the DPA, et cetera, was discussed and how much it converts. So do you have any comments on krill oil versus clary sage seed oil?
I don't have a comment on that. Goal is to focus on DHA levels. I think people get DPA is fine because DPA gets converted, but really you want DHA, like the omega, like the 6 double bond DHA. That's what your body wants for its membranes. Like EPA and DPA are very, very small, minor players when it comes to human physiology. And so focus on the DHA levels, and probably the algae oil versions are probably the best for vegetarians. To be honest with you, you're going to get higher quality DHA from algae. Because krill oil is actually animal based, right? So vegetarians. And so I would go to an algae oil based system for that. And then biggest thing for vegetarians is that you got to keep up. I tell we're not plants. So if you're not going to eat animals, you need to make sure in your diet, you put things in there that you would normally get. So your carnitine, creatine, sulfur amino acids. Okay, you must get those in your diet. You're going to really have negative health consequences. And same thing with fat. Like you need to get good old-fashioned fat into your system. And so, the phosphatidylcholine type lipids you need to get into your system. Phosphatidylcholine is another thing that can really get depleted in vegetarians.
Now we've got a question that is quite complex. Keith asked about the environmental, health, diet, epigenetic factors that lead to autism. I believe autism is the final common pathway of everything goes wrong. As in Alzheimer's, there's multifactorial, and Dale Bredesen has found if you treat the main contributing factors, you can reverse some of the cognitive impairment. So I think autism is going to be the same. It's multifactorial. No two kids are different. And yes, environment plays a role. So your comments.
Yeah, so autism, we know exactly what autism is. So autism is a combination of two things. One, it's a mitochondrial insufficiency combined with an inflammatory environmental stress. You put those two things together. So first and foremost, it is not a genetic disease. Okay, it has genetic predispositions, but it is an environmental disease. The epidemiological increase in incidence is not physically or mathematically possible for a genetic cause. Okay, the human gene pool has not changed that dramatically in 1 or 2 generations. What has changed is that the environmental pressure on these children has gone up. And it could be anything. So what happens now is, imagine all of a sudden the temperature of the planet went down 10 degrees. We all have people that don't mind the cold. Some people can't stand the cold. Some people need an extra jacket. And you're dealing with that from a mitochondrial perspective. So what happens with this increased inflammatory stress, that this mitochondrial insufficiency triggers neural inflammation. Okay, so as soon as the child gets exposed to some sort of triggering event, that neural inflammation goes from an acute event to a chronic event, and they don't resolve it. And what happens in autism then is, once you have inflammation of the brain, once that brain becomes inflamed, the myelination, the normal neurological development of the child, gets disrupted. And that's where the symptomology comes from. So that part, we know in excruciating good detail. Okay, we have, like, there's 36,000 research papers on autism out there. Like, we know this in excruciating good detail. Now, the underlying cause, like what a child gets exposed to, okay, whether he falls off a skateboard and gets hit on the head, or gets a bad flu virus, or gets too many vaccines at once for that one child versus another child, or gets exposed to something else. There's 100 different ways a child can get exposed to an adverse event. And if they are if they have an underlying mitochondrial weakness when that happens, and when they're younger, the younger the brain, the more susceptible they are, because the brain is born with virtually no myelin at the beginning. And so during the myelination, this is why we see the incident rates of autism tail off after age 5 or 6. The brain starts getting more and more myelin, and the more oligodendrocytes that the brain has, the more resilient the brain becomes. So the brain does get more resilient with age in terms of this particular stress. And that's why the diagnostic rates kind of peak in that age 3 to 6 range, 5 to 6. So yeah, so multifactorial, yeah, it's clearly. And if you look at siblings of autistic children, they'll have the same biochemical profile. So they'll very, so mitochondrial DNA is passed maternally, of course. So any kind of mitochondrial weakness is going to be always seen similarly within common families, but the stress might change. So all that stuff we know in great detail. And then the gender bias is based on the beta estradiol level differences in prepubertal boys and girls, because beta estradiol gives girls protective capabilities that boys don't have. And that's why autism is so much more severe in girls. But the all-cause mortality in girls with autism is like 5-fold average rates, where for boys with autism it's around just around 2. So girls who get autism, they always have a more severe case of it. So anyways, long answer to your question. So that's what the whole lecture series is all about. So that's why it's kind of fresh on my mind. I've just prepared all that data, and we have a clinical trial starting, and we just have some dramatic outcomes for children that are taking plasmalogen, like the omega-9 and some mitochondrial support. So like kids waking, they stop their self-abusive behavior. They start playing with they start getting two-way conversations, two-way playing with their siblings. It's pretty amazing to watch.
So I know there's probably questions in the audience about success in reversing the cognitive impairment. For example, on the show, we had Catherine Reed, a biochemist, whose daughter had severe symptoms, couldn't even communicate. So she experimented. And in her child's case, she was lucky. Removing the glutamate from the diet reversed all the cognitive impairment. And the child ended up being very social, etc. A lot of kids might not be that lucky. For example, some autistic kids, their mothers had antibodies against the kid's brain in utero, which is going to be a real problem. So what success have you had in reversing it? And can you tell us anything about reversing the symptoms? What's the success rate? Which kids are more likely to react, etc.?
At this point, we are batting 100%. I think we have between 50 and 100 kids. I don't have a single case study of any child that we've been interacting with. Okay, when we start reducing, the first thing we see is reduction in neurological inflammation. Okay, if we can get the brain inflamed down. The omega-9 plasmalogen precursor, and this has been surprising to us, but I have a doctor in Chicago has one of his patients, autism, head bang for 15 years in a row. Family is, distraught with it. Put them on plasmalogens, some mitochondrial support. the mother sends them a video in less than 2 weeks of her son playing in a water park with his father, okay, that they have these things happening. Like some of these rare diseases. So we are seeing, the question will become how much recovery can be occurred. Okay, we're seeing that when the inflammation reduces, the two-way play, the communication. Our head of manufacturing has an autistic child, and he is continuing to improve with time. So what we do know for sure is if we restore the myelination, the brain can begin back on its developmental path. Okay, that's number one. And that seems to go hand in hand with reducing the inflammation of the brain. So the question really becomes is how much reversal can occur. Okay, so in terms of these acute observations, okay, on average, within 2 weeks, we start seeing the parents coming back to us and saying, wow, this is like a different child. He can go to school now. Like the people at school, the school is telling me, what have you done? He's interacting, he's not violent, he's calmer. And so this brain inflammation thing, I think we have dramatically underestimated how pervasive it has been. And with this plasmalogen precursor, for the first time, we can actually directly address the myelination of the brain, and it's causing the inflammation to go down dramatically. So that's kind of success rate right now. Seriously, it's 100%. Like I don't have a single case study of any of our doctors out there who have started an autistic child. The question is how much of a success rate you're getting, right? Like you're going to get some improvement in social activity, and then they have like the older children when they're 15, 16 years old, okay, that's a much longer recovery path, but they start seeing positive outcomes within a relatively short period of time. And so it's up to us now to fine tune that. So
That is
that's why
so,
the clinical trial starts next month or next week. In a couple of weeks, we have the autism trial begins for everybody. So
that is so important. Going through the questions here, one person wants to know why your formulations have clove oil.
well, for just the omega-3, the DHA, because pure DHA is susceptible to oxidation. So clove oil is a very powerful antioxidant, and so we've used it in our formulation just as people use vitamin E, for example. And so it's part of and it also, clove with a little bit of cinnamon for our liquid formulation is a little more palatable than the pure DHA. But I think for our gel caps, we are moving towards, we're going to remove the clove and cinnamon from the formulation going forward. So we can make it custom if we have to. That's the only reason it's there for. It's a very potent antioxidant and antibacterial.
The person who asked about bradycardia, that's slow heart rate, with a pacemaker wants to know, should her supplements be revised or would they be the same? I mean, obviously it depends on her particular condition.
Yeah, I think you just treat the heart, like you want to get the heart to a situation where it can heal itself. Okay, that's the thing. We are here to create the environment. So when we provide the plasmalogen precursor, the child is healing themselves. We're not coming in as some antagonistic trying to fight something. We're not trying to block something. What we're doing is actually saying, you know what, if we can restore the mitochondrial health, we can restore the oligodendrocytes' ability to maintain myelination in the presence of inflammation, the child will heal themselves. The heart will heal itself. So you need to give it the right environment, right ingredients in the right environment. So in terms of the bradycardia, you want to make sure, look at, if you got all the other biomarkers in place, I would go with the omega-9, the prodrome glia, right off the bat, because that's what the heart sarcolemia will suck right up. Like I would just make sure they're not, and then once that's in there, then let that time do its magic, and we'll see how you can slowly recover from that. Because the human body is very resilient. It has high recoverable capabilities if you give it the tools to recover.
Darshana, if you have a specific question concerning your mother and her condition, please put it in the chat or raise your hand, because I'm not sure if there's a question in there. However, Alfonso, who seems to be very knowledgeable, cites that from the mouse study, does dose-dependent plasmalogen precursor effect for the neuroprotection stop at 50 milligrams per kilogram? That means for glia, 6 servings, 12 caps per day.
Yeah, so typically if someone has dementia right now, my recommendation is a bottle of each a week. They should be on a bottle of neuro during the day and then at nighttime. So yeah, so I would go to, 6 to 8 capsules of each of them and get that restoration up. What we've learned over the last couple of years, first we started really conservatively, start with small doses, go slow and slow and slow. And we've realized, bring it up, get that effect, the patient will feel better faster, and then you can bring it down a little bit later on. But definitely if you have cognitive impairment, seriously, again, cognitive impairment, I have almost no circumstance, okay, unless it's a massive stroke issue or some other really serious biological cause. But if it's an age-associated cognitive decline, even early Alzheimer's, when you get them on the higher dose of plasmalogens, you will see benefits within a relatively short period of time. It's very rare not to. What we found was it was a dose-dependent issue. So back to the dose, yeah, the trial went up to about 50 milligrams per kilogram at that 4 mils per day dose, which is 8 capsules. But no, you can go higher than that. I have a little boy, Thatcher, who has an rare disease. He's probably on the equivalent of, I don't know, 12 or 14 capsules a day in a liquid formulation. So the stuff is very safe and very tolerable. And you just basically bring it up, see an effect, and bring it back down again. But if someone actually has an overt cognitive impairment, then you want to deal with that. And I would get to a rule of thumb is go through a bottle a week for the first month, and you should be seeing significant improvements. Not just in their brain function, but their neuromuscular function and a lot of their aches and pains. The glia is really powerful for sleep improvement. And that's another serendipitous thing that we didn't really realize going in, because it was designed for myelin. It's designed for remyelination. And we didn't really realize the people coming back to us with the sleep improvements. So it's really, I wish I could give you a bunch of complicated stories, but it really is, it's so much of it in your body. Like that's what's interesting about this thing. We're not dealing with some real minor micronutrient. It's very large percentages of it in your body. So your body will suck it up if you're deficient.
Kumar asked, does this help for older people, meaning folks that are already over 70 that have some dementia? I think you've already answered that, but
Yeah, absolutely. Yeah, like my dad's 86. I've had him on the stuff where he functions like he's in his 60s. So absolutely 100%. This is, look, I made this stuff for me and my family, and then I share it with everyone else afterwards. So this is stuff like my entire, like I have family members with bipolar disorder. It's helped them get their empathy back, and weird things. They start cleaning the room up. Like you see strange things happening when these people start getting better. Like it's like this brain inflammation. I really had no prior anticipation of this. If anyone told me that the things that I would tell them they're absolutely nuts. This is not normal for a scientist to see this kind of stuff. And so that's for the elderly people, like you saw the age, the mortality. It's a 30-year difference in mortality. It's just not worth it to be deficient in plasmalogen.
okay. Darshana, why don't you ask your question about diphtheria?
Well, I was wondering, we figured my mother was probably autistic. About a year before she died at age 93 in 2015, she had so many behaviors that really seemed like somebody with Asperger's, but she was so smart, she covered a lot of it. Anyway, when she was a child, she got the diphtheria vaccine, and then she had diphtheria, was very sick, and they gave her some kind of medicine to which she turned out to be allergic, and she swelled up and she was really miserable. And I'm wondering if going through that might have created some kind of extra inflammation in her brain, which turned on the tendency to be autistic.
Yeah. So what happens with inflammation? Okay, people forget they think the brain, like a lot of the part of the development of the brain is in the pruning process.
Yeah.
Okay. And it's the ability to actually reduce neurological, you want a smaller number of strong signals. And the myelination, the best way I explain people what myelination is like being in a busy, crowded room, right? Okay, you're at a conference or someplace, you got a bunch of rooms and you got people having conversations. There's 10 different conversations going on in the room. And you can mentally scan from one, you can tune into one conversation, then you can tune into another conversation, then you can tune in another conversation. Your ability to tune in and tune out is all the myelination. It's a protective coating on your axons. Okay, then you have a group of say 10 people, they're talking and you can hear that conversation. But if one person in that conversation starts yelling their head off, okay, and creating a loud noise, the question is, can I differentiate that yelling person from the other 9 non-yelling persons? Okay, and that's what the strength of the myelination allows you to do. But for an autistic child, what happens then is that if the volume is low, they can hear all 10 voices. But if one person starts yelling, they hear one very loud voice of all of them yelling at once because they can't differentiate the yeller from the rest of them. And that's the myelination process. And so what happens is that pruning process, and that's what we have underactivity in certain brain regions, like the corpus callosum and the white matter regions have underactivity. And in certain areas like the frontal cortex, temporal cortex, you can have extra neurological activity because some of those neurons haven't been, the relationship hasn't been strong and strengthened. And so you have this excessive neurological activity in certain regions. Now, when you have people with high functioning autism, what they've done is they've taught themselves how to isolate the noise. It's like someone who likes to study with loud music. Okay, they can put the music on loud, and that music drowns out everything else, and they can focus on their schoolwork, right? And that's what an autistic child does when he starts banging his head. And there's overt ones, but there's also subtle ones. So how some of these really high functioning autistic kids become so high functioning is that they have learned to focus, okay, on one thing. And that focusing on one thing, this behavioral focusing, they do that to compensate for the noise in their head. And if they get really good at that focusing, they can become, clairvoyant, like they can become super functioning in certain areas, right? And they become unfunctioning in other areas, like what we consider normal, all these social cues, all these subtle things. Being a human and living in a social environment, there's so many little subtle concepts of, taking a hint and getting so they just can't do all that stuff, right? But that's kind of where, so for your mom, if she had an acute inflammation event, depending on how long it lasted, remember, a little bit of damage can sometimes help, right? Because the inflammation creates glutamate. It actually creates an increased activation of the brain. So a little bit of that can actually be stimulatory. Like a bipolar patient, for example, a bipolar patient is at their threshold of activity because of the protein kinase C system, is they're kind of already kind of self-primed, and that's why they become so like their intellectual capacity and their ability to get things done, they eventually burn out, unfortunately, right? They have 12-year lower lifespan than the rest of us, but they for that period of time while they're on steroids, if you will, like they're powerful. And so a little bit of inflammation can actually be powerful. So that's what happens for some people. They get a bad inflammation event, and if they're able to survive it and they're able to kind of get through it, and females are a bit better at hiding it than men, just for numerous reasons. But yeah, that's probably what happened. They had some acute event, and she was lucky enough that it didn't completely destroy her, and that she was able to, fake it for the rest of her life.
Yeah. Around age 30, she actually lost 5 pregnancies, and they gave her medication, which was used at the time. And I heard that she got worse, that the autism behavior got worse. I was born when she was 30. I was the one that survived. Okay. From those experiences, I'd always assumed she felt like she'd failed and she was a bad mother, and so she felt guilty and ashamed, and that's why she got worse. But maybe it was maybe there was more inflammation.
Chronic brain inflammation. It's like a concussion. Like people get a concussion, bad concussion. Okay, that doesn't go away. Like they have an inflamed brain for years and years and years. Like the inflamed brain very rarely goes away. What happens to a concussion when you give someone a return to play designation, They have just learned to adapt to the concussion. They work
Right.
around the concussion.
Right.
They trick their coach into letting them play again. But the inflamed, like the brain is like a castle with a moat, right? Like if something happens on the inside of it, it's got to fix it from the inside. Okay, it's not like getting a bruise on your shoulder where you get the really good circulatory system and you can kind of clear that stuff. And that's
Right.
why these improvement of circulation of the brain, like even the intracranial improve blood flow. And this is where the glycocalyx comes in, right? If you get better blood flow into the brain, if you can get things in and out of the brain better, then you can clear some of that toxicity better. But the brain is like it's concave, like so it's to protect from outside influences. But as soon as a Trojan horse gets into it, okay, it's got to deal with it from the inside. And that's where these inflammatory diseases of the brain, they persist for very, very long periods of time. Like these studies with adult autism patients, like they're in their 50s, and they have the same level of brain inflammation as the 8-year-olds. Like that they've been living with that their entire year, and that becomes normal to them.
Yeah.
And so when you reduce that neurological inflammation, some of these people go, "Wow, this is what normal feels like. I never knew what normal feels like. This is what a normal person must think. They can actually, put their thoughts together." And they stop, and we see reductions in self-medication because they stop seeking all of these mechanisms to kind of help themselves. So I think again, this neurological inflammation, chronic, I think we've just dramatically underestimated it, many aspects of it.
Yeah. Can you get a lot of inflammation from one concussion, like from a car accident where I was only unconscious for a minute or two and I never had a headache?
Yeah, it's like, look, humans are very variable, right? Like it's going to be so, and concussion of the brain is a variable thing. If you got a really bad knock to your head, chances are you still have underlying inflammation. Like the thing is, the brain has a lot of excess capacity. Remember we talk about Parkinson's. We got to lose 80% of our neurons in the substantia nigra to have mobility, to see a mobile difference. Like that's how much reserve capacity the brain has. The brain can work around a lot of damage. Like you can get away with it for many, many years. And that's why it kind of creeps up. Like Parkinson's and ALS are good diseases where you kind of have a toxin that could have happened 10 years in the past, something to knock down 20, 30% of your neurons, right? You'd never even know it. Okay, had a bad, food experience or something with some toxin. And then it doesn't show up until 20, 30 years later when you've had your natural, where your brain has had some over age-related decline. Then all of a sudden, these things start popping up. And the cause of it happened 20 years ago, knocked down 20%. I think that's what makes it so difficult for us to kind of connect some of these dots sometimes. And that's where that one paper I was showing was a Coxsackie virus, which I thought was really interesting. Like where they had two different animal models. One was susceptible, one was not. The susceptible one, that damage stayed down there. The mitochondrial, and this is what we see in ALS. Like we see this mitochondrial insufficiency, it gets knocked down once and it stays down. It doesn't recover. It's the weirdest thing. And so I think we got, anyways, so back to this whole thing. I think we have some of these things persisting. These long COVID sufferers, they just get underwater. They just can't get themselves above water. And that's where I just was on the call with another patient that had really bad myocarditis and pericarditis. She said her heart, and she starts taking the omega-9 plasmalogens, and she said she could actually feel her heart filling back up again. It's really strange. So we're learning. And that's why the network of doctors is important, getting people's feedback. The challenge of, it's multifactorial treatment, right? You want to look at many aspects of human physiology. Obviously, certain things are going to have, bigger influences than others. But in some people, like if you have a really bad B12 deficiency, like it really can, like you can chase your tail for a long time, okay, to find that one thing. And then all of a sudden, wow, we've all heard stories of that, right? Someone goes in for a B12 shot and they're just a whole different person, right? And so we have to realize that kind of, like, sometimes we're just missing that one piece of the puzzle. And statistically speaking, we know which ones have higher probabilities than others, and some will give more weight than others, and they'll contribute to each other. But that's the trick of this, integrated health is looking at the whole body and making sure we don't miss it too much.
Yeah. Thank you so much.
Okay, Paula,
Yes, hello. I'm wondering if taking the plasmalogen precursors have any contraindications for people who are on psychiatric meds.
No, the opposite, actually. They can start coming down on some of them. So for bipolar, and a lot of people, when it depends on the severity of the psychiatric disease that you're treating, of course, right? Like a lot of these drugs are helpful, and they can't, like Zyprexa, for example, or the atypicals, like for hallucinations or psychoses type of thing. My typical approach in these kind of psychiatric diseases is, how do I make the person safe enough to tolerate the drug? Because so they can get the benefit from the drug at the lowest dose they possibly need, and the drug has the least amount of other negative consequences. Because the problem with a lot of the psychiatric drugs is that the benefit comes with quite a bad cost, right? And then if you can start lowering them down, like if you can bring the benzodiazepines down, and all you have to do is treat with, say, a very targeted dopamine blocker for something, then that person can have their control of their life back. So no, there's no contradictions on the psychiatric meds that I'm aware of, and I have many people on them, and so if anything, it helps them bring it down. I've had some people that really, for the anticonvulsants, and they can bring the anticonvulsants down, for example. So,
That's good news.
Yeah.
This is my sister who's in her early 70s, and she's been bipolar for 40 years or more, but she's by herself and doesn't have much support. And I don't think she lives across the country from me, so I'm not there to help her, but does not have a very healthy lifestyle. I don't think she eats well. I don't think she gets much sunshine. So beside taking these plasmalogen precursors, what would be the number one other lifestyle thing that she could improve that would help her?
Well, priority number one is the omega-9, the glia. Okay, start with the glia, because that will have the biggest impact. Like it really calms, like if you can get the brain calmed down a little bit, they can start getting control of life back. Like it's really quite shocking. She'll start calling you back and asking you how you're feeling, right? Which probably has, right? And so that kind of thing. And then the B vitamins, get the B vitamin, because what happens in all bipolar patients, when you have an inflamed brain, it consumes vitamins faster, especially your methyltransferase system, your B12, like betaine, methylfolate. Those are really critical to maintain, N-acetylcysteine, that kind of stuff. But start with the glia, and they usually will take that, because often the bipolars are really suspicious about taking anything, right? And so you want to have something.
She's supplement crazy.
Okay, well then I would start with the glia first. Try to get her on a bottle a week for a little while, and she'll feel her brain calm down. And then make sure you keep up on the B vitamins is the next priority for them. And then at that age group, I try to keep them on creatine. People think of creatine just for muscle building, but sometimes when you can't, if you have patients out there that you're having a hard time getting their homocysteine levels down, you're doing everything by the book, and that homocysteine still just chronically sticks around 12, 14, you give that person some creatine, good old-fashioned creatine, you will lower their homocysteine levels very, very quickly. Because almost a good portion of the homocysteine in your blood comes from the making of creatine in your liver. Your kidney makes the first half, and the second half gets made in your liver, and that's a methyltransferase system. So the two main contributors to homocysteine is creatine and phosphatidylcholine. So those two things, if you're having a hard time, if you just give them those two things, you see their homocysteine levels drop. Because what you've done is you've reduced the body's need. Like so, because your body makes about 3 to 4 grams of that creatine every day. And so if you take it endogenously, what you've done is you've freed up that methyltransferase capacity for other things, and your homocysteine levels will drop. So that's in the elderly, because you're working with frailty and muscle wasting in the elderly, getting creatine into their diet, because they're eating less, right? They're moving less, all that kind of stuff. And we forget
the same thing.
Yeah, exactly. So And those are simple stuff that are really well tolerated for most people.
I imagine she could find a trained specialist in your protocol out in Tucson.
Oh, yeah, I'm sure.
Yeah, I'll look. Thank you. This
Take a
is very helpful. Thank you.
Okay, Atta, your question.
Yes, one more question. And by the way, doctor, it's last year, about last April, I got COVID. And about 3 months later, I developed a hyperglycemia with fasting glucose of about 380. And but with no other symptoms. And was with the declining A1C. Now, none of the, so I was put immediately into the diabetes bucket, which none of the diabetic medications, including high dosage of metformin, work. In fact, it made my case situation worse. Now, I did some testing on my own, and I find out that my somatostatin is very high and epinephrine is very low. And so I came on the, and I started using the alternatives like berberine and vanadine in order to bring my glucose down. Now, about January, I got COVID again, and just last week, I've had a really bad severe case of vertigo, to the point that I had to take the CAT scan and MRI just to make sure that I don't have actually a stroke. So in your opinion, what are the things that the remedies that could be working out, knowing now that COVID probably could be the underlying cause of this?
Have you considered the GAD65 antibodies? You might be developing autoimmunity to your islet cells. Anyway, just a thought.
Yeah, okay, the main driver of glucose levels in your blood is actually your skeletal muscles, okay, unfortunately. So what you need to do is, why glucose gets up in your blood is because your cells, your glycolytic capacity of your cells go down. And when you get COVID, you get inflamed. And when you have inflamed, when your body's inflamed, there's glutamate. So that one person who said that they really helped their autistic child by reducing the glutamate, absolutely a good thing. Okay, so what happens then is if you have an inflammation, this glutamate goes into your cells and it competes for glucose. So your body has a choice, either it metabolizes the glutamate or it metabolizes glucose, okay, or you have to have both. And then your skeletal muscle. So the reason why resistance training improves glucose levels, so the basically lifestyle aspects of type 2 diabetes, is because it's your skeletal muscle that metabolizes all of your free fatty acids, for the most big bulk of it. And your free fatty acid metabolism in the fasting state. So when you're fasting, your fat cells are digesting triglycerides and releasing free fatty acids in your blood supply. Right. And that free fatty acid gets chewed up, like your heart and your skeletal muscle use it. If you can't process your free fatty acids, then those take up your glucose. So your body, since your fat cells are shoving out free fatty acids, if you can't process them, you can't process glucose either, and so glucose will go up. So what you have, the way to get rid of the diabetes situations, you have to increase the glycolytic capacity of your cells. So one is the good old-fashioned, I tell people, N-acetylcysteine, carnitine, CoQ10. Like, make sure the mitochondrial capacity of your cells are up to snuff. And then the skeletal muscle part has a big play on that. So reducing the inflammation will be your big drivers of that. So what COVID is doing, it's causing inflammation, and inflammation creates this diabetes, basically, type 2 diabetes. And so that's where you want to work on that aspect. I would recommend stuff like curcuminoids. They're very potent anti-inflammatories. Make sure your gut health is up. Like I said, the N-acetylcysteine, carnitine, CoQ10, and then the plasmalogens, like, really will just the omega-3, the DHA ones, like, it'll suck that down pretty quickly. And then once your inflammation goes away and restoring your metabolic health, then your glucose levels. Because when people take insulin, forget about, so what happens when you have insulin insensitivity? It's like your body has glucose floating around, right? And it wants to go into cells. And the cells are saying, hey, I'm all full. I'm full here, no room for you. Okay, and the cells say, I'm not taking any of your glucose, right? And so it doesn't let the glucose come in the cells. And then when you take insulin, you're basically saying, well, damn you, you're going to take your glucose and you're going to like it. And so what the insulin does, it just forces the cells to take it glucose, thinking that,
Yes.
right. And so at the end of the day, you have no choice in the scenario, but improving the glycolytic capacity of your cells. Okay, and so that's what you need to improve your mitochondrial function, improving skeletal muscle mass, that type of thing, creatine, those things will all help and bring that inflammation down on those two separate levels, and then glucose levels will come down. And that's why those certain things cure 100% of diabetes. Skeletal muscle mass cures virtually all diabetes.
Yes, I'm actually under regular exercise and high resistance and
Yeah, and I tell people, especially when they're recovering, okay, big thing for recovery is don't do too much. I always tell people exercise is bad for you. Recovering from exercise is good for you. So you want to exercise, but then you need to give your body time to recover from it, because it's the recovery from it that you get the health benefit, not the exercise itself. And so what happens sometimes, and especially we do with pro athletes, and some they overdo it. So what you want to do, you want to stimulate, and then you want to wait, let your body build up. Then you want to stimulate, let it build up, and that kind of back and forth, and gradually notch up and then as your muscle mass grows in a slow way, then that acts as a filter. It helps by increasing your skeletal mass. It's like increasing the size of your liver. Basically, you have much better capacity.
Unfortunately, I brought it down now. My glucose level now, it's about fasting glucose is between 150 to 200, which is much better than the 380 and plus. And but it's slowly coming down.
What's your fasting triglycerides?
Fasting triglycerides, actually, I didn't test that.
Okay.
But I know when the first time when I did that,
triglycerides?
everything was in a normal level.
Well, normal is bullshit. Like it needs to be under 100. Okay, it needs to be under 100, and you want to check your HDL levels. You want to get your HDL levels over 50, hopefully around 60. The other thing you want to take is phosphatidylcholine, because you want to be able to. The other thing that drives your cholesterol, your glucose, is your membrane cholesterol levels. It changes your glucose absorption rate, and so phosphatidylcholine and the DHA plasmalogens will help that reverse cholesterol transport system flow.
So you think your supplement would help?
Sorry?
Do you think your supplement helps in this case?
Oh, absolutely does. The neuro, it really just, there's huge data, not just from me, in terms of reverse cholesterol transport. So the trifecta is typically, if people that are healthy, they'll have if you get triglycerides under 100, your HDL over 50, those people will typically have good plasma, as a general, good peroxisomal function. Okay, that's your kind of your trifecta you want to have. And then, but the triglycerides are a big indicator of your metabolic capacity, and keep those under 100.
And do you think that the vertigo is now could be another long hauler's symptom?
Absolutely. So as you restore your plasmalogens and the white matter part, like the vertigo period of your brain inflammation, also make sure when you're getting better, there's a little thing you can get this, your eustacia tubes, right? People forget about these simple little things. Like you can make sure that you can buy a little thing from the drugstore. It's a little puffer that puffs in your nose. Make sure you get your eustacia tubes opened up, because if that's not open and you're not clearing, okay, that can have in your hearing, like getting your ears, making sure that your ears are working properly. But a simple little thing of, because what'll happen if you get into a sickness, people do this when they get into, say, muscle wasting or an acute disease. Sometimes their eustacia tubes will collapse a bit, okay? And so it's actually quite, there's simple solutions to that. You can just look for a little, I think it's called eustacia or something like that. It's like, sometimes we forget the simplest solutions can actually have benefits for us, but make sure that your ears are all
okay. Very good. Thank you very much.
You're welcome.
We have a question in the chat about prevention. So can this be preventative for folks?
It's even, it's most powerful as a preventative. Absolutely. Like that's like even in all of our animal studies and all that kind of stuff. And that's the #1 priority is prevention long-term. Yeah.
Okay, this question, I think, has various answers depending on the individual, but somebody asked, what are the symptoms of a concussion that occurred decades before?
That's a tough one. Like, well, if you take a look at, if you do advanced brain scan, for example, like they don't get them done on a regular MRI, but you want to look at your white matter microstructure. So if you do an advanced brain scan, you can look at the quality of the myelin wrapping. And so that's one way to look at it. Like you can look at, it's called your radial diffusivity. Like, so there's advanced technology called NODI, like neurite orientation and disbursement technology. So there's some advanced MRI that can look at the white matter microstructure. The take home is like, and I saw the soccer thing there, but yeah, if you have a kid playing football or hockey or soccer, I'd be giving them the glia. The protum glia is basically your concussion preventer. Like if you have that glia in your brain in advance of a concussion, your brain can maintain its myelination rate in the presence of the injury. So yeah, absolutely. If I had kids playing contact sports, they would not be going on the field without taking plasmalogens about 2 to 3 hours beforehand. I'd be dosing them up every day before they go on the field.
Any other questions? Put it in the chat or raise your hand.
Yeah, family members, exactly for that. I saw that. So my aunt, for example, she got so bad, she's in a long-term care facility in Canada. And my uncle, she's in her 80s now. My uncle's also in his 80s. And she got so bad that she didn't even recognize her son's name, like my cousin. And and he was not letting me know what's going on. So I found out and I started shipping him the neuro and the glia. And within about a month, she started coming around. She now goes out for dinner with him. She can recite everybody in her 50th wedding anniversary album. She knows where everybody is. She is communicable. Yeah, we see things like this, like really serious recovery. And I just did a podcast with another group, and someone came on, told me their experience about their mother, and she started giving her mother was in long-term care. And her birthday apparently was on March 27th, and she was very aggressive last month or so. And then the caregiver went in, and the mother asked the caregiver, "What day is it?" Remembered that it was her daughter's birthday, started singing "Happy Birthday," and they recorded it and sent it to her daughter. Like these, like it's crazy. It's not normal. Like you don't think of it this way, but we are seeing pretty in our family members. Like we have a lot of family members that we're all affected by this stuff. So yeah, so for sure.
Well, unless there are any other questions, I want to thank you profusely because this is fascinating and very helpful over a large range of conditions that are affecting many of us. Our next presentation will be April 18th at 11 o'clock in the morning, California time, where Mayor Snyder will come back with more tidbits on how to improve vision naturally. And then on May 2nd at 11:30 in the morning, California time, we will have Tim Jackson come and give us a talk. We're open to any speakers that you recommend and any last questions before we sign off. Otherwise, I will edit this and we'll put it on our website. Oh, and your slides. Some people expressed an interest in your slides.
Yeah, I kind of put them together pretty quickly for today, so there was a lot there. So yeah, I'll send the slides to you. If you can find me a folder, it'll probably be too big to send by email, but I'll send it to you guys.
Okay.
Awesome. It was a pleasure meeting all of you, and I look forward to seeing more of you guys in the future.
Thanks, Doc.
Okay. Cheers.
Thank you.
You're welcome.
Okay, thank you so much.